All right, we've got a little bit more to wrap up with enzymes. Enzymes work at best under certain environmental conditions, and especially temperatures and different pHs. So if the pH is increased or decreased, or the temperature's changed, and this most of the time we associate a rise in temperature, then these molecules can be rendered useless, and because their structure changes. We talked about protein structure in chapter 3 as well, and we talked about denaturation. And so what happens is that if for instance, the pH is decreased, this can affect the hydrogen bonds in the active site, and once the active site becomes unraveled, it does not recognize the substrate that it needs to recognize, and therefore it doesn't catalyze the reaction. So the reaction goes slowly, and when that happens, the cell dies. Some cells do have helpers. These are small molecules that are attached to the enzyme, and if that molecule is not attached, then the active site unravels and it doesn't recognize the substrate. And so these enzyme helpers can be two different types. There's cofactors. Cofactors are usually metal ions or complex inorganic molecules, right? Where coenzymes are types of cofactors that have complex organic compounds that they're derivative of vitamin. And so if they don't have those enzyme helpers, then the reaction is not catalyzed. The next part of the chapter deals with membranes. We talked about phospholipids being the major component of membranes in chapter 3 as well, and the phospholipid has a head that is the phosphate group head that is hydrophilic. It likes water. It also has two hydrophobic tails. These tails do not like water at all. And so if you have a bunch of phospholipids and they're exposed to water, what they will do is they will make a sphere, because they will arrange their heads in the direction of the water and put their tails away from the water, just like circling the wagons, right, in an old western. But what happens is when you circle the wagons and make this layer, what you trap on the inside is water. So you have water on the outside, you have water on the inside, and in order for a membrane to form, then it has to make another layer of phospholipids with their heads facing the inside of the membrane. And so when you see this, it's like magic, all right? Sorry. There we go. And so when the membrane is formed, water's on the outside, you see the orange heads, right? And their little tails are facing toward the inside of the membrane. When the sphere is formed, water is trapped on the inside as well. So what happens is they have to arrange their more phospholipids to where their heads are pointing toward the inside of the cell, and therefore those tails are facing the inside of the membrane. And this is really important because the inside of the membrane, that inner part, with the tails is very hydrophobic. So if water wants to move into or out of the cell, it cannot do it just through the membrane alone, and therefore any other polar molecule. Likewise, if you had a nonpolar molecule that wanted to get in, and it would not be able to get in because the heads are hydrophilic, water loving or polar loving, and so a nonpolar molecule would have a hard time getting through the membrane as well. So what happens is there is this thing called the fluid mosaic model that describes what membrane looks like. All right, so we know it has these phospholipids, and they're the fluid part because they are so hydrophobic and hydrophilic. If you punctured a membrane and then pulled whatever you used to puncture the membrane out, those phospholipids would just move right back and cover that hole because of their water loving and water fearing makeup. All right, and so you have these phospholipids, and they're fluid. They're not like in concrete. It's not like a concrete wall. And then the mosaic model, part of it is, mosaic is a piece of art that's got different kinds of art. You use kind of different kinds of art. So the mosaic part of the membrane are the proteins. There's proteins attached to or embedded in this bilayer. All right. And so probably the most important of these is called the transport protein. It's a protein that allows molecules that are polar to go through, into and out of the cell. And it works like a door on a central hinge. If you push the door on one side in, then the other side of the door rotates out, right? And so the Three Stooges are waiters at a fancy restaurant. They have this kind of door in their that goes into their kitchen. Mo goes in and pushes the door open to go in, and Curly's standing there at on the other side of the door. And as it's pushed open, as he pushes, the other side of the door comes out and hits Curly and knocks him down, and he drops all the food, etc. And then the eye poking starts. These allow for these water-soluble substances to move through and out of the bilayer, and so they won't work unless that transport protein, those particular molecules won't be able to get into and out of the cell without these transport proteins. The receptor proteins are those that bind messages, and then they send those messages into the cell, and the cell then, the DNA, the nucleus then decides to take that information and act on it. And so receptor proteins trigger, for instance, changes in mitosis when cell division occurs and things like that. So receptor proteins are those that are like the satellite dishes that are out there recording information and sending it back to the nucleus for action. Recognition proteins are those that recognize cells being of like or different in a cell tissue layer. Okay, in the case of the white blood cells, they have proteins, recognition proteins associated with them called antibodies. And when a foreign cell is in the bloodstream, the antibodies attach to the cell and see if they recognize those same antibodies. If they don't, then they recognize an antigen, which is a recognition protein on the cell that's not supposed to be there, then the white blood cell attacks the red blood cell. The foreign cell and tries to get rid of it. Right? Adhesion proteins are those that are sort of associated with the surface of the membrane, and they help attach cells in the tissue layer together to form a tissue. We talked about cell junctions in animal cells and the adhering junctions, and this is the protein that helps allow for those cells within the tissue layer to stick together. Lastly, what we're going to talk about, and in your book, I think it's the very first thing that this chapter talks about, is the movement of molecules. Remember, we said that their molecules are in constant motion, all right? And as they are moving and colliding with other molecules, what's going to happen in some cases is there's going to be a concentration of molecules that is greater in one area of the cell than in others, right? And this is called a concentration gradient. And so one region of a fluid contains more molecules than the neighboring region, right? And so if there is a concentration gradient that exists, then molecules will want to move from an area of high concentration to an area of low concentration. They will move down their concentration gradient. They don't need energy to do this. They will automatically move from an area of high concentration to an area of low concentration. Now, once they are equally distributed between the two fluids, then there is no concentration gradient, and therefore you have equal molecules. They're not going to move down that concentration gradient because there's not a concentration gradient to move down. So they want to evenly disperse between the two fluids. So if you have gaseous concentration gradient, all right, or two fluids that are not separated by a membrane, then the molecules move from an area of high concentration to an area of low concentration until they're equally distributed, and this is called diffusion. All right. ATP is not needed, their energy is not needed for this to happen. All right. But if you have a membrane that separates the two fluids, then that's a type of diffusion called passive transport. Right. It's just diffusion through a membrane. All right. But no ATP is needed because a concentration gradient exists, and the molecules move through one side of the membrane into the other down a concentration gradient. If I were to go out and get a dead skunk, roadkill skunk, and I bring it into the lecture hall, and I place it in a waste basket, all right, and then I let it just sit there, the stink molecules would be really concentrated in the waste basket. But as the time would go on, those stink molecules would start making their way out into the room, and until that at one point they're all equally distributed in the room. But the ones that are sitting close to the the wastebasket will smell the stink molecules a lot quicker than the one they're sitting furthest away. But at some point in time, all of the students in the room would be able to smell the stink molecule, and that's because they're moving from an area of high concentration to an area of low concentration. Diffusion. So we have passive transport through, which is diffusion through a membrane. If we have water moving through this membrane, then we call it osmosis, all right? And it's responding to a concentration gradient, moving from a fluid that has a higher concentration through a membrane into an area that has a lower concentration. When we talk about osmosis, a lot of times we talk about tonicity. Tonicity is the relative concentrations of solute in two fluids that are separated by a membrane, right? So we're going to look at a red blood cell, right? A red blood cell has a membrane. It's in plasma, all right? Plasma has a solute in it, salt. So does the red blood cell, right? If they have equal amounts or percentages of salt, then both fluids are said to be isotonic. If they're isotonic, then there's no net gain or loss of water in either of those fluids, all right? But if there's a differing amount of concentration of salt in the red blood cell than in the plasma, then water is going to move in a direction to help dilute the water to where it's equal to the amount of water inside the red blood cell. And so if there is a differing amount of concentration, let's say inside the red blood cell there is a 10% salt solution, and then outside in the plasma there's a 5% salt solution, all right? So if it's a 10% salt solution, that means that 90% of the solution is water. If you go out to the plasma, that means it has a 95% water content in the solution, right? And so it has higher water, right, than inside the red blood cell. So the one that has the higher concentration of salt, in this case the red blood cell with 10%, is hypertonic. Sorry. The plasma, which has the least amount of solute, and therefore is the hypotonic solution, right? The hypotonic has more water, the hypertonic has less. So what happens if you have a hypertonic solution and a hypotonic solution that are separated by a membrane? Water will flow always from the hypotonic solution to the hypertonic solution. All right, water flows from hypo to hyper. So in the case of the red blood cell is hyper, the plasma is hypo. So water from the plasma will go into the red blood cell to try to dilute the solution equal amount as out in the plasma, and so the red blood cell swells. If it was reversed and the red blood cell had a 5% salt solution and the plasma had a 10% salt solution, therefore the plasma is hypertonic, the red blood cell is hypotonic, then water would move from the hypotonic to hypertonic, and so we would have water leaving the red blood cell and going out into the plasma, and the red blood cell will shrink or shrivel. Now, water can't just go through the plasma membrane by itself. It needs a transport protein, and so this specifically any type of diffusion through a membrane by way of or via a transport protein is called facilitated diffusion or facilitated transport. It's helped by that transport protein, right? Again, this is a passive transport. No ATP is required. Lastly, though, sometimes the red blood cell needs, the cell itself, it could be the red blood cell, but a cell might need more of a solute than the diffusion itself can provide, right? Sometimes it needs to hoard certain molecules. And so if the concentration of a particular molecule outside the cell is less than inside the cell, the molecules that are inside the cell will want to move out, right? They want to move out into the naturally, but what the cell wants to do on some occasions is to hoard or store more of those molecules than it really needs at that particular time. It's going to use, but so a concentration gradient exists, but instead of the molecules moving from an area of high concentration to an area of low concentration, the cell moves molecules against their concentration gradient from an area of low concentration into an area of high concentration. And this movement of molecules against their natural way, that down that concentration gradient, if you're moving it against the concentration gradient, then energy in the form of ATP is required, right? So an example is a magnet. If you put two south poles together, you have two magnets, you have two south poles, and you try to put them together, you'll feel the repulsion of those two magnets. They do not want to come together, right? Naturally, it's just against natural law. So what you can do is take those two magnets, and if they're not powerful enough, you can actually put those two south poles together, but you have to use force. You have to force them, and that's like molecules moving against their concentration gradient. They don't want to move that way, so you have to use force, and that force is energy in the form of ATP. If you turn the magnet back around and you have a north pole and south pole, you put them together, they instantly go together, right? And so that's like the regular concentration gradient and the movement of molecules down that concentration gradient. They'll do that no matter what. You don't have to add energy. But if you want to move those molecules against the concentration gradient, then you have to use ATP. All right, so that's chapter 5. We will use some of the terms that we learned in chapter 5 to go on to chapter 6 and 7, which are cellular respiration and photosynthesis.